Great. So thank you so much for that generous introduction and for the opportunity to speak with you all today. So as David mentioned, just recently started my lab 2 years ago at the University of Washington. We call it the Systems Neuroscience and AI Lab, or SNAIL. And we're interested in the intersection of machine learning and neuroscience, broadly speaking. We think a lot about modeling and analysis of neural population recordings at the level of single trials, computation through neural population dynamics, communication between brain regions in brain-wide recordings, and learning plasticity in the brain, thinking about how populations of neurons change their activity as the brain learns something new, oftentimes thinking about the learning rules that might support those types of changes in population activity, and often using brain-computer interfaces as an experimental paradigm because of some kind of exciting tractability features that it offers for thinking about learning. And so today's talk is going to be more focused on the top 3 bullet points. And I'm going to focus on this paper that we recently presented at the most recent NeurIPS, Active Learning of Neural Population Dynamics using two-photon holographic optogenetics. And so this was a really fun, exciting, highly interdisciplinary collaboration between a couple groups, my group and Kevin Jamieson's group at the Computer Science School at the University of Washington, and the Allen Institute for Neural Dynamics with Kayvon and Carol here also in Seattle. And this work was really led by Andrew and Lou. So Andrew recently finished his PhD at UW in Kevin Jamieson's group, focusing on active learning theory, and is now a postdoc at Berkeley. Lou was a postdoc working in my group, focusing on modeling population activity. And she recently moved to start her own lab at Georgia Tech. I'll be discussing some data collected by Martin and Kayvon at the Allen Institute, with many thoughtful conversations with Matt Bull and Carol Svoboda, who directs the Allen Institute for Neural Dynamics, and I understand spoke with you all a few weeks or months ago, and has really been quite supportive of this collaboration. I also want to point out Joe and Christina on the right here, who kind of came onto this project after we had submitted this paper, but have really taken the lead on modeling and experiments moving forward now that we've lost Andrew and Lou to their next steps. So setting the stage, we and many others have observed that neural population activity evolves lawfully over time. So if we record the simultaneous activity of, say, a few hundred neurons, we're often thinking about this activity in a neural state space, where each axis indicates the activity of one neuron, and a point in the space indicates the activity across the entire population, okay, at a particular moment in time. So we can really only visualize this in 3D, representing 3 neurons, but actually the dimension of this space is the number of recorded neurons. So it's a high dimensional space. This is one point indicating the population activity at one moment in time, and as time unfolds, these points, these neural states, tend to evolve lawfully over time as a neural trajectory. And the neural population dynamics govern how this trajectory unfolds over time, and how these neural trajectories can be interestingly different across trials or conditions or tasks. So these dynamics are often very structured, such that if you look at these neural trajectories from many different repetitions of an animal performing some behavior, many different single trials, then the neural trajectories tend to reflect some low-dimensional dynamical motifs. So in this case, the neural circuit gives rise to an attractor motif. So all these trajectories lie in a 2D plane embedded in this 3D space. And those trajectories are attracted to a stable fixed point in the neural population dynamics. So other examples of these dynamical motifs include a line attractor, a saddle point, an oscillation, and these are all simple linear dynamics. So they can all be described by a linear system, which will be important for some of the modeling we'll talk about later. And these simple motifs are interesting because they can often be the building blocks of richer dynamical computations. So one reason that many of us are so interested in population dynamics and these types of motifs is because they've been implicated in potentially implementing neural computations in many areas, including motor control, motor timing, decision-making, working memory, social behavior, learning, and this is just to kind of name a few examples. So there's a very rich history of fitting dynamical models to neural population data, and then analyzing those models to try to understand the computation being performed by those dynamics. And so the traditional approach there is to first record some population activity while an animal performs a task of interest, and then fit a dynamical systems model to those recorded neural data. Okay, and this really relies more or less on passive observation of the neural population. And by passive, I mean you're imposing some task, but otherwise you're just kind of passively observing the neural activity in the brain as it unfolds. And this will be in contrast to active observation that I'll be talking about more throughout the talk, where we want to kind of probe and punch the neural circuit actively to see how it responds and potentially get a fuller picture of the neural population dynamics. So this passive approach suffers from two key limitations. So first, the inferred structure is purely correlational. So it can't really be interpreted with any notion of causality. Okay, so for example, dynamical structure may be apparent due to the task design, how your stimuli are presented, due to unobservable internal states that have their own time courses and may be difficult to control experimentally. And so just because your model learns that state A tends to be followed by state B, that doesn't mean that A causes B. Okay. And then so second, the experimenter has very limited control over how the neural population dynamics are sampled, because we're just passively observing the circuit. And so this can lead to inefficient data collection. So you may inadvertently be oversampling some parts of the dynamical landscape while undersampling or maybe entirely missing other important parts of the landscape. So in this work, we really wanted to address these two limitations. And our goal was to figure out how to actively design the causal circuit perturbations that will be most informative to a dynamical model of the population activity. So there are two key elements here. We want to first actively design our data sets. And second, we want to do that using causal circuit perturbations. Okay, so by actively designing our data sets, we hope to be able to fit a good model using less data, using less experiment time. So experiments are, of course, resource intensive. That's motivation, I think, on its own. But you could imagine that recording data to fit a model might just be one component of your experiment. So less time on that component could free up more time in your experiment for those other aspects. So for example, we're very interested in studying learning and plasticity. I'm not sure, Carol may have discussed this project with you all. And so we often in that project find ourselves wanting to identify and probe the circuit dynamics before and after we ask an animal to learn a task. And we would love to be able to free up more trials for the animal to work on the learning component of the task by spending fewer trials probing the dynamics before and after that learning. Okay. And then so second, these causal external manipulations of the circuit activity are going to allow us a causal interpretation of our model. Okay, so we've been thinking about this using circuit perturbations based on two-photon holographic optogenetics to photostimulate mouse motor cortex, while simultaneously recording neural responses using two-photon calcium imaging. So just to kind of sketch out the talk for today, I'll start by kind of going more deeply into the experimental setup. Then I'll talk about some low-rank auto-regressive models of population activity that we've found are kind of surprisingly useful in describing these data sets. Then I'll describe some active learning theory and an algorithm that we developed using it to learn these auto-regressive models using less data. Okay, and I'll show a demonstration and some results of that. And so full disclosure up front, we haven't tried to design actual mouse experiments online in closed loop yet, but I will show two demonstrations in relevant online and offline data-driven settings that I think serve as an exciting proof of concept that motivate kind of that future work. And I'll close by describing that. So launching into the experimental setup. So as I mentioned, we're using simultaneous two-photon imaging and two-photon optogenetic photostimulation. And so this photostimulation is going to utilize holographic optogenetics to afford temporally precise cellular resolution optogenetic control over ensembles of the very neurons that we're imaging. Okay, so we're imaging using GCamp6M in these experiments as the calcium reporter. This is in mouse motor cortex. We image a 1 millimeter by 1 millimeter field of view. And so I'm showing you an example image frame on the right here. We are getting yields of in the high hundreds or low thousands of neurons. So in this image, each of these red annotations are regions of interest that Sweet2P has identified as cell bodies in this image. So the stimulation protocol that we use in the data that I'll describe today is we deliver stimulation pulses that last 150 milliseconds, and we space those pulses by roughly 1 second. Okay, and each time we stimulate, we apply one of 100 unique photostimulation patterns. And so that pattern is a hologram that simultaneously targets 10 different neurons. So it focuses light onto 10 disks. Each of those disks is 10 microns in diameter, about the size of a cell body. And we have some constraints, so our photostim optics currently require those stimulation disks to be co-located in roughly the same kind of quarter of the field of view. So we can't stimulate any 10 neurons in the same frame. They have to be somewhat co-located, but we can cover the whole frame across different stimulation pulses. Okay. And all of this is going to be while the mouse is engaged in just undirected spontaneous behavior. So there's no task. And we'll come back to that at the end. And so using this protocol, we've amassed these data sets that contain about 2,000 trials in total. So each trial is one of those stim pulses plus the relaxation second spacing. And so those 2,000 trials contain randomly interleaved 20 repetitions of each of those 100 different stimulation patterns. Okay, so I know that was a lot. Let's see what this looks like in action. So here I'm showing a video of the photostimulation, and these white discs show, the white circles show spots that we're photostimulating, and the yellow is indicating increases in activity of the population relative to photostimulation onset, and blue is indicating decreases in activity. And so you can see that these stimulation patterns are kind of co-located in a particular kind of quadrant-ish of the field of view. And since there's kind of a lot going on here, I want to step through kind of a couple frames and just point out some of the interesting features. Okay, so we see both what I'll call direct and indirect responses to this photostimulation. So by direct, I mean, if we put light on a neuron and it responds, I'm going to call that a direct response. Okay, as opposed to if we don't put light on a neuron, like over here, and that neuron responds, I'm going to call that an indirect response. And that's going to be due to connections between the stimulated neurons and that neuron that we did not apply light to. Okay, we can't say much about how many kind of synapses are between these neurons because of the temporal precision of the calcium imaging is a bit too slow for that. But because we're causally perturbing these cells, if there's a reliable response in an untargeted cell, we know that there's some sort of causal pathway between the targeted and untargeted cell. Okay, and so these direct responses are relatively reliable. You'll see some cells respond more than other cells, okay, and that can be due to the amount of opsin expressed in the different cells. But overall, these direct responses are relatively simple, relatively easy to model. You put light on a cell and it responds, and then that response decays. And so that's kind of the less interesting modeling component for us. And what's much more interesting is modeling these indirect responses, because we think that those are kind of very telling of the population dynamics, right? How things are connected. And we see a really interesting heterogeneity of these indirect responses. So I'm putting some green diamonds around some indirect responses to kind of highlight them, to show that they can be kind of spatially near or far from the stimulated neurons. Okay, they can be excitatory, shown in yellow, increases of activity, or they can be inhibitory, blue, decreases in activity. And they can be more or less reliable, indicating quite a bit of trial-to-trial variability that likely depends on things like the internal state of the circuit at the time of each stimulation, so ongoing dynamics in the circuit. Okay, and these responses can be kind of spatially specific, or they can be spatially broad. Okay, I'll just kind of thumb through a couple more frames to give you kind of a flavor of what some of these indirect responses look like. Okay, so these are kind of the basic data sets that we're going to try to model and build up this active learning framework around. And so now I'll transition to tell you about some of these models that have been kind of surprisingly effective at modeling these data. So we're just going to be talking about linear autoregressive models. So here I'm going to use X to denote the population activity. So if we're recording n neurons, this is going to be an n by 1 vector. So if it's helpful to have a number in your head, you can think that n is, say, 1,000. Okay, and so then there's just an input, u of t, which is also going to be a n-dimensional vector. And we just have we encode the photostimulation just as a binary vector. So ones and zeros, just indicating whether or not each neuron was directly stimulated. And then to model the dynamics, we have a square n by n dynamics matrix A and an input matrix B that is also n by n, and then this offset vector V. Okay, and so here is one example neuron, traced out in blue the activity of that neuron. And then I'm showing photostimulation that was to that particular neuron with these pink pulses. Okay, and you can see when there's a pink pulse, the blue jumps up. Okay, and this is in orange, the predicted activity from an autoregressive model like this. And you can see that it kicks up as well to track the blue. And there's also shown here some of the indirect photostim. So here is a moment when other cells were stimulated, but this neuron responded, and the model was able to capture that indirect response. Okay, and there's another example of that over here. So we find that adding a few lags helps these models out a little bit. So we have ARK models in general. We find that K of 4 tends to work roughly the best in these data sets. And so basically that just means we have 4 different lags of this equation with 4 different sets of these parameters, A and B. Okay, and then we add what will be a critical design element here, which is we impose some structure on these A and B matrices such that they are diagonal plus low rank. So they're the sum of a diagonal matrix and a low rank product. And this is kind of motivated by the observation that we and others have ubiquitously found that dynamics tend to be low dimensional. Okay, and this will be really key in deriving efficiency gains through an active learning approach. Okay, so the first trace I was showing was actually a full rank model. And down below, I'm showing the same exact trial, but modeled with a rank 35 model. So the low rank term is rank 35, and you can see that the predictions are very, very similar. So it's capturing more or less the same amount of variance as the full rank model. Okay, so how do we use these models in an active learning framework? So our goal is, given at a particular moment in time in the experiment, given whatever data has been collected so far, we want to design the next set of stimulation patterns that will be most informative to improving that model, and then we'll apply those stimulation patterns next. And the motivation here is we want to minimize the amount of data needed to learn this predictive model. So the approach that we use is called Fisher information maximization. So Fisher information is a measure of the sensitivity of a model to changes in its parameters. It quantifies how much information and observation carries about the parameters of your model. And so by choosing to collect observations that are most informative, we'll need less data to fit that model. Okay, and so this is a decades-old approach stemming from the 1950s in statistics that we've brought forward and applied in the context of this low-rank estimation problem. Okay, and so rather than applying this technique to the ARK model itself, we actually apply it to an even simpler linear model that we call this causal connectivity. Okay, and so this causal connectivity model is basically trying to just model the kind of integrated impulse response of the neural population to a particular input U. Okay, so this is summing over ideally infinite time the neural response as a function of an input U that goes through some matrix H, assuming that your neural population is at some steady state at time 0, and that input is applied just for the first time step, and there's 0 input otherwise. Okay, and so ideally we would sum over infinite time, but we really don't need to do that because these stimulation responses decay quite quickly on the order of a second or so. So we can, in practice, truncate this to that time scale. Okay, and then ultimately what this H matrix is measuring is this causal connectivity, which is if I stimulate neuron I, how much response integrated over some short window should I expect in neuron J? Okay, and so actually we apply this Fisher information maximization to this simpler, basically linear regression problem in the case of a low rank constraint on this H matrix. And there's a very intimate link between this H matrix and the auto-regressive model, because if your data actually come from this auto-regressive model, you can just write out this H matrix as a relatively simple function of your A and your B parameters. Okay, and just to see this intuitively, if you don't have, if K is 1, so it's just the single time step auto-regressive model, then your H is just going to be B. Okay, and if you have more lags, then A starts to play a role. Okay, so when Andrew cranked through the active learning theory, he derived this key result which states that the precision of estimation of this matrix H, which is the inverse of the estimation error, okay? So that scales according to the sum of these two terms. So the first term quantifies the amount of photostimulation input that's directed into the top R right singular vectors of H, where R is the rank of that matrix H. Okay, so those top right singular vectors, those are the input dimensions that produce a lot of output. Okay, and then the second term quantifies the amount of photostimulation that is directed isotropically, so spread out uniformly across all input directions. And these two terms are not weighted equally. There's an n minus r here and an r here. So again, r is the rank of that matrix H, and n is the number of neurons. And so n tends to be much larger than r. So we might have 1,000 neurons with a rank 35 model. And that means that this term is kind of quite a bit more important than this term. Okay, so this theory motivated the following active learning algorithm, where first we initialize some model by basically collecting some random photostim data. So we pick B photostimulation patterns, apply those. So those are just chosen uniformly to kind of spread out in the space. We don't know anything about the system yet. So then we apply those photostimulation patterns, we record the responses. We then fit this ARK model to the resulting data, and then we derive that causal connectivity matrix, which I'll call H hat, from that fitted model. Okay, and so now we have kind of a very small number of data points estimated H, so it's not going to be perfect. And so now we just want to kind of continually improve it by using the active learning concept. And so basically to cover both of those two terms, we have some hyperparameter P. So with probability P, we'll sample from kind of the active part. So that is picking photostimulation patterns that optimally span the top R right singular vectors of our estimated H matrix. Okay. And then otherwise, we'll sample points, sample photostimulation patterns uniformly. Okay. And so this is basically to say early in the experiment, we don't have a good estimate of H yet. And so it's important for us to kind of be a bit random rather than focus lots of energy on dimensions of a kind of not so well informed estimate of the matrix. Okay. And we use P as a hyperparameter here rather than setting it to kind of n minus r versus r scaling, because this theory here is supposing you know the true H, okay? But of course, at the beginning of our experiment, we don't know the true H, we just have this estimate. And so it's really kind of going to be problem specific how much energy is kind of best spent randomly versus actively throughout the experiment. Okay, so after we have kind of chosen a set of photostimulation patterns, we apply those photostimulation patterns, we record the responses, we fit the ARK model now to all the photosim response data collected cumulatively thus far in the experiment, and use that to update our estimate of the causal connectivity matrix, that H hat. Okay, and then we just rinse and repeat. We do this over and over again. And there are some other hyperparameters here, like how many stimulation patterns do you pick at each kind of iteration of this loop. And there will be issues around computational constraints and timing of the actual system that might determine if you want to kind of fit models more frequently or less frequently, applying fewer new simulation patterns or more simulation patterns informed by each update of the algorithm. Okay, I'll show demonstrations where we're choosing basically 200 different patterns in each step. Okay, so let's get into seeing how this algorithm works. So I'll start by showing how it works in a data-driven simulation, and then in kind of a real data setting that's a little bit closer to an online experiment, but is actually offline and is kind of the best that we could do without being online in the rig for now. Okay. So here we fit a data-driven simulator of mouse motor cortex by basically taking an entire 2000 trial data set and fitting an ARK model to that. And then we treat that data-driven model as a surrogate for mouse motor cortex. So basically we have this simulator now. We can apply arbitrary photostimulation patterns to that simulator, whether they occurred in our data set or not, and how well it will generalize to unseen stimulation patterns is an important question. So this is obviously not going to be as accurate as it would be in the mouse brain itself, but we think it's an important step forward. And so when you apply those photostimulation patterns to the simulator, you'll get some neural responses out. And so basically you can treat this like a fake mouse brain that you can just probe with various stimulation patterns and try to estimate the underlying dynamics and this causal connectivity matrix. And because this is a simulation, we can compare our parameter estimates with ground truth in the model. Okay. And so when we do that, we see that our model performs kind of substantially better than, so here in red is the estimation error of our active algorithm, looking at a mean square error in the values of the parameters of that H matrix relative to baseline algorithms that would either randomly pick stimulation patterns that have kind of the same joint statistics as the actual stimulation patterns that we apply in the experiment, or uniformly spread out patterns in green. And so overall, what you're seeing here is that our model is able to exploit the low rank structure in the simulated dynamics to learn a better model with fewer samples. So it's able to use less data to fit kind of the same accuracy of model compared to these non-active baselines. Okay. And so again, the intuition here is because the dynamics are low dimensional, then it's kind of a waste of experiment time to have stimulation inputs that are expected to push in directions that are outside of those dimensions captured by the dynamics. So really, we just want to focus our effort in pushing the system around in the low dimensional space where the dynamics live. Okay, so here's a second demonstration of the technique. And so here we're using the data sets to kind of simulate an experiment in a different way, where we're saying, okay, we have this whole data set, but let's not observe it. Let's treat it as kind of trials that we can choose whether or not to request those trials, and then fit our models based on only the trials that we've requested so far. And so this kind of mimics what would happen in an active closed loop online experiment, with the important constraint that we can only request stimulation patterns that were actually applied during the actual experiment. And that's a huge constraint because, as I mentioned, there were just 100 different unique photostimulation patterns applied in these data sets. And the space of possible stimulation patterns, if you're choosing, say, 10 cells at a time from a population of 1,000 cells, that's an absolutely enormous space that is dramatically undersampled by just 100 patterns. Okay, but nonetheless, we tried it, and it shows a bit of an improvement for the active learning. And so what this looks like is, first, we just hold out 20% of the data set and just put that aside for cross-validation. Okay, and then we apply our active algorithm to iteratively request a specific batch of the held-in trials. Okay, so first we're going to start by randomly choosing a bunch of trials, and then we're going to fit a model, and then we're going to use the active algorithm to pick which of the remaining held-in trials we want to observe next, 200 at a time. And so each time we're observing new trials, we're refitting our ARK model with all the trials that we've requested thus far. And then we evaluate the fitted model on the held out trials at each iteration. So throughout our kind of estimation process, as we're getting more and more data, each time we can evaluate the fitted model on all of that held out data set to kind of track how we're doing in terms of generalizing to unseen test data. And so those evaluations will be done here using mean squared error on the neural responses. Okay, so here we can't, of course, compare to the ground truth parameters because we don't know the ground truth parameters that generated the real data, but we can ask how well does the model predict the neural responses that we did observe in that held out data. Okay, so just to orient you to this figure here. So here, this is the number of trials that we've requested, and then how that impacts the mean squared error on the held out trials. And we should expect these two curves to intersect and overlap at this final evaluation point, because based on how we've designed this experiment, at this point, both our active model and a random model have seen the entire set of held-in trials. Okay, so they're fit to exactly the same data. They're going to make exactly the same predictions, have exactly the same mean squared error. Okay, but earlier on the iterations, what you'll notice is that there's an improvement in the prediction error for the active model. It's a modest improvement if we're looking just at the scale of the vertical axis here. But we think it's quite impressive nonetheless, because as I mentioned, this is an extremely constrained setting. This algorithm is only allowed to choose from 100 candidate stimulation patterns. Actually, it's 80 because we're holding 20 of those out for cross-validation. Okay, and that spans a tiny fraction of the space of possible stimulation patterns that we could choose from in a real experiment. And even so, what you can read out from these curves is that the active algorithm, say at 1,000 trials, is performing about as well as the random experiment does with 1,600 trials. Okay, and so to us, that demonstrates a really promising improvement in data efficiency that we were after all along here. Okay, so to conclude, I hope that I've convinced you that active learning can accelerate learning of the neural population dynamics. Okay, we have shown some gains in data efficiency, and these gains are largely due to the low dimensional dynamics. Okay, so we're spending our stimulation budget preferentially on stimulating the kind of in manifold directions and wasting less time stimulating out of manifold directions. Okay, and so far we've demonstrated this in this data-driven simulator and this active ranking experiment where we request trials in a different order from these real data experiments. Okay, and then thinking forward, I had mentioned earlier that in these data sets, the animal is just doing spontaneous behavior, so not actively engaged in a task. And so an important question to ask is, what if the animal is engaged in a task? And it may be that these simple linear models can effectively model the spontaneous activity, but they might be less effective if the animal is engaged in a task, because task-driven behavior tends to induce kind of larger fluctuations in the neural activity, potentially richer, potentially nonlinear dynamics. So we may need to think to nonlinear models in some future work. So we're thinking about that. We're of course thinking about now building this active learning framework into online closed loop experiments where we can run the algorithm with the mouse in the rig and say, okay, these are the next patterns I want to choose. Apply those patterns, measure the responses, refit the data, suggest the next patterns, and so on. And scientifically, I had mentioned we're really interested in using techniques like this to study learning, to probe the dynamics, to measure the dynamics before an animal learns a task, and then measure them after the animal learns a task, and see whether there are interesting changes in the dynamics, and see kind of what types of changes are possible and what types of changes are maybe more difficult. And can we think about the learning rules that drive those types of changes? Okay. And lastly, since we're doing all this modeling of perturbations and circuit perturbations, and since we have models that seem to do a pretty good job of capturing both this direct and these indirect photostimulation responses, I think it really begs the question of, can we use these models in closed loop in the rig to actively control the neural population dynamics? So if we know how the population responds to different stimulation patterns, we ought to be able to choose the stimulation patterns that our model will predict will drive us to certain interesting parts of the neural state space. And I think kind of the sky's the limit in terms of the scientific questions that one could probe if you had really spatially and temporally precise closed loop control of a population of neurons. And so with that, I will thank the many colleagues, students, postdocs, collaborators that helped facilitate this work, and the various funding agencies who've supported our work. And I'd be happy to take any questions.